Hi, my name is Marina Rosenlund, and I am presenting on environmental toxicants in breast milk of Norwegian mothers and gut bacteria composition and metabolites in their infants at 1 month. So the bottom line up front was to determine whether exposure to multiple environmental toxicants that are present globally result in a change in gut microbiome composition and function in 1 month old babies. As technology advances, more environmental toxins will be present in the global population. We still don't know how these affect people, as a lot of these technologies are new, so it is important to determine the effects of these toxins on babies aged 6 months or less at as this window is where the gut microbiome is established. So, for background, why breastfeed? It provides antibodies against viral and bacterial pathogens and intestinal parasites. It decreases chances of developing inflammatory diseases such as diabetes, cardiovascular diseases, obesity, and various cancers. Breast milk plays a unique role in primates because of the slow development of our immune system. We already receive antibodies from placenta during gestation, but breast milk provides precise protection against pathogens. It also supports the growth of healthy enteric flora, and it helps the mother recover after birth and promotes emotional bonding between mother and child. While nursing, oxytocin is released, which helps reduce the size of the uterus and lessens postpartum bleeding. It is also cost effective, convenient, and eco friendly, and it contains a lot of nutrients, including IgA, lactoferrin, lysozymes, bifidus factor, oligosaccharides, milk lipids, and milk leukocytes. So, what happens if a child is exposed to toxins contained within breast milk, and why do we care? Persistent lipophilic chemicals are stored in the mother's breast tissue over time in the fat. This is not just during pregnancy, but throughout her life they can accumulate. DDT is an environmental toxin which has been found in nearly all human milk tested since 1951. Additional toxins such as pesticides and metals such as mercury, lead, and arsenic exist in breast milk as well. Studies show greater levels of toxins lead to greater risk for negative developmental effects. And prevention measures should be developed to prevent future developmental delays in children. So, to summarize the article, how do environmental toxicants present in breast milk affect the gut microbiome composition of 1 month old infants? How do these toxicants impact gut microbial metabolism, more specifically, the production of short chain fatty acids? Does the level of exposure to environmental toxicants influence the level of gut microbiome alterations? And what bacterial taxa are most affected by toxicant exposure? So, in this study, there were 267 mother baby pairs in Norway. Breast milk samples were collected 1 month postpartum to measure environmental toxicant levels, and infant stools were collected at the same time to determine gut microbiome composition. Mothers and babies who had had antibiotics 2 weeks or less prior to this study were eliminated completely from the study to not present any confounding variables. 28 environmental toxicants were measured and quantified using high resolution mass spectrometry. Polychlorinated biphenyls are chemicals used in industry and are banned in many countries. Polybrominated diphenyl ethers are flame retardants. Polyfluoroalkyl substances are found in food packaging and nonstick coatings, and organochlorine pesticides are known environmental pollutants. These are all toxicants studied within here. Um, For the microbiome analysis, the DNA was extracted in the V4 region of 16S rRNA, which is a highly conserved region, was amplified using PCR, and then sequenced using Illumina MiSeq. Short chain fatty acids were quantified in stool using gas chromatography mass spectrometry. Um, the Spearman correlation test was used, which detects associations between toxicant levels and microbiome shifts. False discovery rate prevents false positives, and linear regression is controlled. It controls co-founding factors. So in figure one, we see in block A the measurement of PFAS toxicants in low, medium, and high exposure levels. There are significant differences between low and high concentrations with a p-value of 0.01. And also significant differences between medium and high exposures with a p-value of 0.003. There was not a significant difference, or not statistically significant difference between medium and low concentrations. This graph indicates higher exposure is associated with shifts in the microbiome composition. Section B correlates with PFAS, and there's some. In this plot, each dot represents an infant's microbiome, and they are color coordinated in the same way, with low exposure being green, high exposure being purple, and medium being orange. There was not significant segregation of the dots in this graph. So variability is still high among individuals. In part C, it measures the PCB, the toxicant PCB 167. Um, and there were significant differences between low and high, but not significant differences between medium and high exposures. Not, and there were also not significant differences between medium and low. Um, there were some overlapping colors, but there were also some clustering of colors more than in B. But this still signifies variability in individual microbiome responses. But this graph, this figure overall, shows high exposure associated with shifts in microbiome composition. In figure 2, the y-axis has the bacterial genus, family, and order, as well as the associated toxicant. The x-axis has the relative abundance of bacterial taxa in the gut. Green represents the low exposure infants, and the purple represents the high exposure infants. The diamonds are the outliers, and the p-values are on the far right, and they're all statistically significant in this figure. So this shows the correlation between bacteria and the toxicant that they were testing. We can see that lower we can see lower lactobacillus in high exposure of PCB 105, PCB 118, PCB 194, and PFOA. Lactobacillus is a beneficial bacteria for gut and digestion, so this is a negative result of the toxicants. We see a higher level of streptococci and high exposure in DDT and oxychlorine. And then we see a lower um, lower Villanella in high exposure individuals. And this is also a negative effect because this bacteria is involved in lactate metabolism. PFOA also resulted in higher enterococcus in high exposure infants. So overall, from this, we see a negative correlation between um, bacteria levels and toxicant exposure, where we see the good bacteria, the ones that are positive in the gut and help support a healthy metabolism, being lower in high exposed individuals, and then negative bacteria being higher in high exposed individuals. In figure 3, they are measuring infants with high toxicant, medium, and low exposure levels in A, and this shows a statistically significant correlation between the two. This is shown to this is using the Shannon scale, which is a scale that measures both um microbial diversity and evenness. You want high diversity typically and high level of evenness. Um, we see in high exposure, um, there is actually a lower overall Shannon score, which a lower number means less diversity or less evenness or both, and then a higher number means um, a higher microbial diversity. In part B, we see the effects of the metabolism and the functions within these microbiomes. Um, Infants with high toxicant exposure have a lower relative abundance of genes involved in the carbohydrate metabolism. This suggests a potential reduction in the gut microbiome's ability to process and utilize carbohydrates, which could affect energy production and digestion. So this is a very negative impact of these toxicants. Um, and then the relative abundance of genes involved in cell motility, which is on the bottom of part B, um, it indicate is lower in high exposure groups. So this indicates that toxicant exposures reduces bacterial movement and colonization ability, which potentially affects the microbiome stability and function. So based off this graph, we see that higher toxicant exposure leads to less diversity within the gut, and it also reduces the important and beneficial functions that these bacteria play within the gut microbiome. This figure shows the relationship between toxicant exposure and short fatty chain acid production in the infant gut microbiomes, and specifically examines acetic acid on the left and propionic acid on the right. It shows that toxicants affect SCFA production. You can see the toxicants listed on the Y column. Um, it PCB 209 resulted in about a 15% reduction in acetic acid. Um, and other toxicants also resulted in a link to a decrease in propionic acid. However, some toxicants resulted in an increase in propionic acid levels, more specifically the toxicant PFOA and PCB 167. Um, Toxicants like PCB 209, PBDE 28, and PFOA show consistently and statistically significant associations across all models. Um, some toxicants or other toxicants in this show wider confidence intervals, meaning they had larger error bars, meaning their effects are more uncertain in terms of their correlation with these different acids. This multiple exposure model usually has smaller confidence intervals, suggesting better estimates, and it helps identify key toxicants while reducing noise from correlated exposures. So the results. Toxicants were detected in 96% of samples. PBDE 154 was only found in 11.2% of samples, so it wasn't as common, but there was a high cross correlation between OCPs and PCPs, meaning they were more commonly found and linked together. There were low correlations between PBDEs and PFASs. Um, the high PBDE 28 and PFOS exposure are associated with lower infant gut microbiome alpha diversity with the Shannon diversity index of 2.3. Like I said, this quantifies microbial richness and evenness. So this is on showing a low microbial diversity level with as the Shannon diversity index has ranges from 0 to 5, 5 being the highest diversity and 0 being the lowest diversity. So this was on the lower side, which showed the correlation between toxicants and uh negative effects on the diversity of the gut microbiome. High toxicant exposure was associated with lower levels of lactobacillus and streptococci as well as veil veillonella. We saw this in one of the figures that I showed you. And PFOA is associated with increased enterococcus and lactobacillus. So while we saw kind of contradicting results with lactobacillus specifically between different toxicants, um, there was still an overall negative correlation with toxicants and microbial diversity and function. So for the metabolism results, we saw higher PCB 209 exposure linked to lower acetic acid levels, and PBDE 28 exposure had a 24% decrease in propionic acid. PFOA and PCB 167 were associated with increased levels of acetic and propionic acid. So the reason these decreases in these acidic acid levels is negative is because acetic acid is used to make phospholipids, other lipids, steroids, fatty acids, and protein acetylation, which are important for humans. And then propionic acid is important and it is negative to have a decrease in levels because it influences the gut barrier function, immune responses, and it can impact other microbiomes throughout the body, including the gut-brain axis. Um, so these toxicant exposures resulted in a significant level variation of SCFA levels, but overall had a negative effect. So What we found from this is that researchers discovered that environmental toxins significantly altered microbial function and diversity during babies' critical microbiome development. This indicates the need for preventative measures and for governmental policies to reduce the exposure of pregnant and nursing mothers to environmental toxicants, um, as these microbial shifts could have long-term negative effects on children's health. So, conclusions. Toxicants in breast milk suggest a decrease in microbial diversity and function. The notable toxicants were PBDE28, PFOA, PFOS, and PCB 167, which had the most significant alterations of the microbiome. It's unclear whether there are health implications from toxin-induced alterations in the microbial composition. As of right now, there's not enough research to understand how these toxicants will affect kids in the future. There are studies showing that it has negative effects developmentally and negatively affects the immune system, but as microbiomes need to be studied more in general, this is one thing we still need to figure out. Um, Additionally, this research was done in Norway with 267 mothers. So it needs to be replicated in different populations and different countries with different and higher contamination levels. So for the questions, could a decrease in gut microbial diversity of infants exposed to higher levels of PFOS and PCB 167 impact their long-term health, such as increasing the likelihood of developing inflammatory diseases in the future? Should public health officials advise pregnant and breastfeeding women to avoid these environmental toxicants to protect their infant's microbiome? What factors other than exposure to environmental toxins might contribute to the varied microbiome results? Are there ways to reduce toxicant exposure to infants through breast milk without stopping breastfeeding? And how can parents or scientists increase microbiome diversity in infants with high toxicant exposure levels? Could the mother take a supplement to change the composition of her breast milk, or are there other ways she can do this? And here are my references. Thank you.